Good afternoon and welcome everybody to our first webinar in 2026. I particularly would like to welcome all our new curriculum students who are 79, as I just was informed, which is an all-time high. This is fantastic. Our today's expert is Professor Cornec Le Gall. Emilie Cornec Le Gall is Professor of Nephrology at the University of Brest and Chair of the Department of Nephrology at Brest University Hospital in France. She is the coordinator of Brest National Center in Rare Kidney Diseases and the co-lead of the French Orphan Kidney Disease Network. Also, currently, she acts as chair of the Genes and Kidney Working Group of the European Renal. Professor Cornec Le Gall's research focuses on the clinical and genetic characterization of polycystic kidney disease, with a special interest in atypical ADPKD. The GENKID cohort, an observational cohort of ADPKD patients from 28 centers in Western France, initiated in 2011, has highlighted the importance of such non-PKD1 variant types in ADPKD severity as well as prognostication. Emilie Cornec Le Gall has contributed to the identification as well as the clinical characterization of atypical ADPKD forms associated with novel cystic genes. Today, Professor Cornec Le Gall will give us an overview on this topic. We are very grateful to have you all here today and have you here, Emilie, for this presentation. Short housekeeping rule: if you have any questions, please put them in the box at the side of your screen, and we will read them out at the end and we'll try to answer. Emilie, thank you for having you here today.
Thank you very much, Jens, for this very nice introduction. I would like to start by thanking ERKNet for the opportunity to speak today about atypical forms of ADPKD and minor genes. I would like to apologize also for not having my webcam on, but we've been really trying hard with IT to get access to the webcam. It doesn't work, unfortunately. Maybe it will work at some point during the meeting. So here are my disclosures. And I would like, oops, yeah. So, as you know, ADPKD is one of the most common inherited kidney disorder, affecting up to 10% of the individuals undergoing renal replacement therapy. And while median age at kidney failure and need for initiation of renal replacement therapy is around 60 years, ADPKD is marked by a strong clinical variability, notably in terms of progression to kidney failure, with at one end of the spectrum, so I will use this maybe, I hope you can see my pointer. So at one end of the spectrum, very severe antenatal forms of PKD, while at the other end of this spectrum, some individuals who may only develop a small number of cysts, and you can barely see them, with well-preserved kidney function, like in this 70-year-old lady here. And so we have this very broad spectrum of disease severity. PKD1 and PKD2 are the major genes in ADPKD, encoding polycystin-1 and polycystin-2, which are two glycoproteins functionally expressed at the primary cilium. PC1 here has 61 N-glycosylation sites, and PC2 has 5 N-glycosylation sites, and you will understand why I mentioned this later on. But the level of correctly N-glycosylated and correctly cleaved and correctly folded PC1 correlates with disease severity. In the past few years, several genes, mostly involved in the N-glycosylation pathway in the endoplasmic reticulum, have been associated with atypical forms of ADPKD or ADPLD, or dominant polycystic liver disease. And also in the past few years, some gene encoding ciliary proteins likely to be involved in PC1 trafficking have also been associated with the PKD spectrum, and we will mention those later on. So polycystin-1 dosage plays a central role in ADPKD pathogenesis, and PKD severity is inversely correlated with the amount of functional PC1. And I was thinking of a continuum in terms of disease severity a bit earlier. One of the explanation of this variable disease severity in some cases might be the co-inheritance of two variants and different alleles. And so in this case, having, for example, two loss of function alleles in trans is embryonically lethal, while on the other end of this severity spectrum, having only one hypomorphic allele of PKD1 might be associated with very mild presentation. And there is a full range of possibility between these two extremes. For example, the co-inheritance of a loss of function variant with a mild hypomorphic allele that might also result in a very early onset form of the disease. And so several modifying factors modulate disease severity. They can be inherited, they can be environmental. We won't spend a lot of time on these aspects because we will focus today's talk about minor genes, but I thought it was important to give some general information on disease variability. The GenKist cohort was initiated 15 years ago by Professor Yannick Lemeur in Brest, so my mentor in Brest, and it's an observational cohort of ADPKD patients. So all the patients who are evaluated in private or public or semi-private centers of this large western part of France. This can be academic centered, but mostly also local centers. So really the idea was to catch the broad variability of the ADPKD spectrum. And so standardized clinical information as well as imaging and genetic information are collected. And we will speak a lot about the GenKist cohort in this presentation. And so as of January 2026, approximately 4,100 patients from 29 different nephrology centers. What GenKist helps us to better understand were first the importance of the genotype-phenotype relation to explain disease severity in ADPKD. It was well known since the late 90s that PKD2 patients do have a milder course than PKD1 patients, and GenKist study allowed to highlight the fact that there were three main subgroups of patients. So these are really broad categories: patients with PKD2 variant, patients with truncating variant of PKD1 who have the more severe course, and patients with non-truncating variant of PKD1 who have intermediate severity of disease. And so this is true at a cohort level, but this is not very informative at an individual level, because you can see that with this renal survival curves that there is a lot of variability. So in order to use the genetic information for prognostication in individual patients, we developed now 10 years ago the ProPKD score, which combines simple clinical variables with genetic variables and ranges from 0 to 9 points, and allows to predict and stratify disease severity in ADPKD. I just, in the introduction, also wanted just to briefly speak about Mayo ADPKD classification, imaging classification, because we will mention it quite often during this talk. You know that typical ADPKD, so mostly caused by PKD1, often PKD2 are classified in class 1. So, you know, it's really enlarged polycystic kidneys with a really classical presentation, and patients are stratified according to 8 adjusted TKV at a given age, and so this allows to stratify patients from 1A to 1E, and patients being classified as 1C, 1D, 1E are considered as higher risk of progression. What I wanted to highlight here is the class 2. Also, so patients in the clinical course, usually 90% of the patients are classified in, as Mayo class 1. Patients with atypical imaging presentation have to be classified as class 2, and you will have two subgroups. 2A will be for patients with either asymmetrical or unilateral or lopsided. Lopsided is when you have 5 cysts accounting for more than 50% of the total kidney volume, while class 2B would be patients with atrophic polycystic kidneys. And it's important to mention that patients in class 2 are more likely to have other genetic etiologies than PKD1 or PKD2, and that there is no prognosis value of TKV in patients classified as class 2. And you can have all the information here, but I thought it was important to give this reminder because we'll speak about when we will describe the different phenotypes, we will refer to this classification. My view on how to characterize someone who has ADPKD and how to assess his or her prognosis is that imaging and genetics are, of course, not mutually exclusive. And it's very important to have an integrated approach and to get the full picture of the disease presentation. And this includes, of course, kidney function, eGFR decline, genetic information, the pro-PKD score, and the total kidney volume, and what is the Mayo imaging classification. And this is an interactive dashboard that was created by one of my co-workers. So now going to the main part of the talk, we are here today to discuss the minor genes and minor genes and atypical forms of ADPKD. My question here is, do you know which of these patients has a classical form of ADPKD caused by either PKD1, PKD2? You can see that they all have bilateral cysts. Some of them do have a positive family history, and in fact, none of them has a classical form of ADPKD. This lady here, she has a type 1 orofaciodigital syndrome. The 3 other individuals, they do have genes, they do have variants that are in genes that have a very different, that are very different than PKD1 and PKD2. And so my short message at this point was bilateral cysts are not always classical forms of, are not always classical ADPKD. And I hope I will provide compelling evidence during this talk that genetic characterization is important. To try to have a better idea of how much so-called minor genes or other differential diagnosis accounted for ADPKD or ADPKD-like presentations in clinical cohort. We had a look in the Jens cohort. This is Dr. Marie-Pierre Audrezet, who is a molecular biologist in Brest and who did all the work in the genetic characterization of patients referred to our center in the Jens cohort and from external centers. And so I will present here some of the results. So this is the situation over the years ago. You know, traditional sequencing was used, and then next generation sequencing arrived, and the approaches and the size of the panels were refined. And we are working on a panel of 27 genes now, and whole exome sequencing is performed when the panel does not identify a class 4 or 5 variant in a gene that is causal for the phenotype. Briefly, so each time the genetic results are analyzed in the context of the clinical and the radiological information, we'll see that it is quite important. And so we report only likely pathogenic or pathogenic variants in gene considered to be causal for the disease. And so here is the situation. So this is a work in progress. We are still updating, but the percentage are quite up to date. And this work was conducted by Guillaume Guyat and Nicolas Zagorek in our center with Marie-Pierre Audrezet. What you see here is that, of course, PKD1 is roughly 2/3 of patients, PKD2 18%, and other etiologies is 6%. So 6% of the patients were considered as having ADPKD or ADPKD-like presentation to have, in fact, a variant in a gene that is not PKD1, not PKD2. Oh, sorry. So among those, we will discuss the so-called ADPKD spectrum genes. And we also do have quite a lot of phenocopies, and we will go through some of the main phenocopies later on in this talk. And so one of the most common minor ADPKD gene is IFT140. It's the third most common form of ADPKD, so after PKD1 or PKD2. And in different clinical cohort, it's usually 1.5 to 2% of the patients. So IFT140 is not a new gene in ADPKD. It was long known to be associated with so biallelic IFT140 variants cause the IFT140 associated ciliopathy. So it's an autosomal recessive disease with a very severe phenotype, including retinopathy, including specific skeletal features, and kidney failure in childhood. Now, what the group of Peter Harris showed in 2022 in a collaborative study to which we participated also was that monoallelic variant of IFT140, and more precisely monoallelic predicted loss of function variants of IFT140 cause a disease of the ADPKD spectrum. The diagnosis is usually around 50 years of age. The phenotype is quite mild, and after this initial description, we recently tried to get a larger view of the phenotype by including a larger number of patients, and so we recruited through different European centers patients with ADPKD IFT140 and ended up with a cohort of 75 patients from 61 families. And what we can say, and I just wanted to emphasize the fact that it is a clinical cohort. So these patients tend to have a more significant presentation than when they are identified in general population. These are patients who are referred to nephrology center, but in this cohort, you can see that they still do have quite a good kidney function there. So you have here female in red, male in blue, so slightly lower eGFR values, but you can see that kidney failure is very rare and usually in patients with other comorbidities. However, after, and I'm just missing the lines here, but after 60 years of age, 78% of the patients would have an eGFR of under 60 milliliters per minute. So they do need some nephrology follow-up, but the presentation is quite mild and way milder than, for example, in PKD1 in red here or PKD2 in blue, you have the IFT140 patient. When you look at the imaging presentation, it is quite distinctive. You can see that patients tend to have a small number of large cysts. So they would be classified, most of them, as having Mayo imaging class 2A, 90% of the individuals. So some do have enlarged kidneys, but it's really important to look at the imaging and do not use just the TKV to assess disease severity. When you see that they are in fact classified as 2A, this is usually associated with quite a good prognosis. They do not have liver cysts usually, or a very small number, and some did have intracranial aneurysm, but it's difficult at the scale of this cohort to really make a strong argument for a specific association. At least when we performed a phenotypic enrichment study at the scale of large cohorts such as 100,000 genome, UK Biobank, there were no association with vascular phenotype, which again does not exclude this possibility either, but the question remains open. So what is really important to understand is that the penetrance of the cystic phenotype is quite incomplete. Not all patients will develop a lot of cysts. Probably if they get imaging later in life, we would find cysts in a majority of the patients, but they would not necessarily develop this significant polycystic presentation. Sorry. So moving on to another ciliopathy gene that was rediscovered in ADPKD, and this is this time a very rare cause of ADPKD. I would like to mention NECate. So again, NEK8 also is a gene that is well known in nephrogenetics, because biallelic variants of NEK8 cause a severe syndromic ciliopathy with renal hepatic pancreatic dysplasia, which is often embryonically lethal. And what colleagues from the Netherlands showed now a couple of years ago is that monoallelic variant in a specific domain of NEK8, in the kinase domain of NEK8, can cause early onset forms of ADPKD. So in the first paper published, most of the cases were in fact due to a specific variant of NEK8 in the kinase domain, which is arginine 2-tryptophan and amino acid 45. And this variant and specific variant in this domain likely affect a dominant negative effect. So here, for example, here, this is a pedigree. You can see that this lady here reached kidney failure at 48 years of age. She had a daughter who reached kidney failure at 1 year, and this daughter subsequently had a daughter who reached kidney failure at 6 years of age. Why such difference in terms of age at kidney failure? Because the mother had the variant as a mosaic, so the variant occurred after the zygote formation and is not present in all cells, and hence the disease severity was a bit milder. But typically in this monoallelic NEK8, kidney failure occurs during childhood, and you can see here another example of a male of 16 years of age with enlarged polycystic kidneys who reached kidney failure 3 years after. So in this paper, they presented 21 affected individuals from 12 pedigree, and with one variant that was identified in 10 pedigrees. There is no evidence for a specific extrarenal manifestation. And so in terms of what is the underlying mechanism, we do know that the mutant NEK8 is well, when so what they did is that they recreated the variant identified in patients, so in cellular model, and that ciliogenesis occurs correctly, and the mutant NEK8 localize to the cilium. However, you can see this here, that the polycystin 2 was not detected on the cilia. There was also a reduced kinase activity and activation of DNA damage response. But the idea here was that because of this mutant NEK8, the polycystin complex could not traffic correctly on the cilium. Now, exiting the cell and the cilium and moving on to a broader category of genes associated with either polycystic kidney disease or polycystic liver disease, I would like to say a few words about N-glycosylation. So what we saw in the past 9 years is that glycosylation associated cystic diseases is an expanding family, and this figure represents the endoplasmic reticulum and the N-glycosylation pathway. And if there are some geneticists, pediatricians, people taking care of patients with congenital disorder of glycosylation in the audience, you might be very familiar with these steps. But this, what we can see here is the nascent polypeptide entering in the ER here via the translocon, and it will be immediately primed by the oligosaccharide transferase on the on several sites with oligosaccharide chains. We spoke about the importance of N-glycosylation for maturation of polycystin-1, and it's the case for many secreted or membrane proteins. And so these glycan chains are plugged on the nascent polypeptide by the oligosaccharide transfer. And the formation of this N-glycan chain is a multi-step process occurring first on the outer side of the ER, then on the inner side of the ER. And you can see that all the steps are very important because at the end, your N-glycan chain must be 3-glucose, 9-mannose, 2-N-acetylglucosamine. This will be first dolichol linked and after asparagine linked after transfer. And this is important because this will allow quality control and correct folding. And so you will see your protein moving on to the different step of protein folding, going through glucosidase 1 here, glucosidase 2 here, and then the calnexin-calreticulin cycle. And each time a step occurs correctly, a little glucose residue in green will be chopped. And then eventually your correctly folded protein will be exported to the Golgi. And if it's not properly folded, it will be it will re-enter the calnexin-calreticulin cycle, but eventually be targeted to degradation if it fails to correctly to fold correctly. So in aggregate, having a complete well-formed N-glycan chain is essential for proper quality control of protein folding and for protein maturation. And so the alteration of protein folding and quality control caused by pathogenic variant of GANAB was a first example of an etiology of ADPKD-like phenotype. So GANAB here encodes the alpha subunit of the glucosidase 2, and the other subunit of this enzyme is encoded by PRKCSH, which is PLD gene. And GANAB loss was shown to be associated with the loss of the mature form of polycystin-1, and the absence of detection here of membrane PC1 in GANAB deficient cell was also demonstrated, and showing that GANAB loss disturbs PC1 maturation and localization. And so a few words about GANAB phenotype. First, the renal prognosis is excellent. Most of the patients conserve a good kidney function. Patients show usually few renal cysts, sometimes large cysts. So again, your Mayo 2A imaging presentation. The liver phenotype, again, however, can be more severe, as you can see here in this patient. And intracranial aneurysm were reported in some pedigrees. Larger cohorts are still needed to confirm this link. GANAB is here. There are other genes very important in the formation of the N-glycan chain this time, which are ALG8 and ALG9, that can be associated with either polycystic kidney disease or polycystic liver disease. But I would like to emphasize that the disease penetrance is not complete. And so ALG8 was initially associated to PLD, but it might can present with predominant PKD, and sometimes in some individuals with very small number of cysts. There is a strong disease variability, but usually the risk of kidney failure would be extremely low and more likely caused by other factors. ALG9, again, overall milder than classical ADPKD. There are some cases of kidney failure reported before age of 60, but it is quite rare, and the determinant of those remains to be elucidated. Most patients would have a very mild presentation, and liver cysts are less common in ALG9. They might be present in some patients, but there are no cases of symptomatic PLD, unlike in ALG8, where you can have symptomatic PLD. Sorry. So I will show here a clinical vignette. This is a 21-year-old male who was evaluated in the team of Sir François Jouret in Liège. And you can see that he has normal kidney function. He has a large kidneys classified as Mayo 1E. And in fact, genetic analysis initially did not find any variant. This was performed in Belgium, and he was referred to our lab to see if we would find something else. His mother has only 1 or 2 small cysts per kidney. She has normal kidney function. My question was, would you ask for genetic testing in this patient, and would you start on tolvaptan for interest of time? We did not discuss the therapeutic aspect. As you, most of you know, tolvaptan is a treatment specific to ADPKD indicated in case of rapid progression or risk of rapid progression. And so in short, what was found in this patient was loss of function variant in ALG9. In this patient and in his mother, there were no variant PKD1, PKD2, including in deep in chronic regions. And so the diagnosis in this patient is ADPKD caused by ALG9, and there is no evidence of benefits in these patients. And I would be very curious to see is MRI in 10 years from now, because I'm not sure that what we know of volume progression, disease progression in PKD1, PKD2 patient do translates to ALG9 patient. This is quite impressive for a 20-year-old male, but we have no evidence of impact of tolvaptan in these patients. Recently, together with Peter Harris in Mayo Clinic, we examined the disease presentation and penetrance of ALG8 and ALG9, taking advantage of our respective clinical cohorts, but also of population cohorts such as Genomic England's and Mayo Clinic Biobank. And what we showed is that the ALG8 and ALG9 are indeed associated with mild forms of ADPKD, that patients with ALG8 and ALG9 variants are likely to develop cysts, but the penetrance is incomplete, and including within families. And most of these patients did not have relatives known to be affected by ADPKD. And so ALG8 and ALG9 are cystic kidney or liver genes with limited penetrance, and these patients tend to have a good renal prognosis. Another gene I would like to speak about is TNAGB11. This is the first family in whom we linked DNAGB11 with the PKD spectrum. I was at that time a postdoc fellow in Peter Harris' lab. And this lady here had an imaging for another reason. She had a metanephric adenoma, she had an MRI, and multiple small cysts were seen. And what was interesting is that her mother was CKD stage 4 at age 78, and she had multiple cysts. And so an exome sequencing was performed in the mother, the daughter, and the brother, and led to the identification of a variant in DNA, GB11, and this was a missense variant, as you can see here. So why did we find this interesting initially? It's because DNA GB11 is the main cofactor of BIP, which is the main chaperone of the ER, BIP, also known as GRP78. And what does DNA GB11 does is that it takes the unfolded protein, brings them to BIP, and together they take the unfolded protein through the folding pathway within the ER. And so the variant identified was right in the middle of a very conserved motif of interaction, so the HPD motif, which allows the interaction between DNA GB11 and BIP. And indeed, the loss of the DNA GB11 is associated with a defect in PC1 maturation. We examined a few years ago in a larger cohort, again, thanks to the collaboration of many colleagues internationally. So the disease presentation in DNA GB11 patients, you can see here that they tend to develop kidney failure, and in fact, if you see them before age 50, most of them would have preserved kidney function. But in this clinical cohort, again, most of the patients after age 50 to 60 did have significant CKD, and median age at kidney failure in this cohort was 75 years. Here we can see a few examples of imaging presentation. So you can see that the renal cysts do appear later than in classical ADPKD and can easily be missed by ultrasound. Here, a female, 35 years, EGFR 99, and she has only 4 small cysts on the left kidney, visible on the MRI, and on the ultrasound, no cysts were detected. Therefore, diagnosis imaging criteria developed in classical ADPKD, ultrasound-based diagnosis criteria should not be used in any of the non-PKD1, non-PKD2 form of ADPKD, and this is very important to keep in mind. In more advanced stage of the disease, we generally observed atrophic polycystic kidneys, except in one patient here of this cohort who had enlarged kidneys. And unlike PKD1 or PKD2 ADPKD, kidney enlargement does not precede renal function loss, and prognosis imaging criteria developed in ADPKD should not be used in DNA GB11 patients or in any non-PKD1, non-PKD2 patients. Now, another gene I would like to speak about is ALG5. A couple of years ago, 3 years ago now, we identified ALG5 as an ADPKD spectrum associated gene. So the story starts with this 56-year-old woman who had bilateral kidney cyst, and a significant family story because 3 half-sisters had reached kidney failure between 60 and 70 years of age. Whole exome sequencing led to the identification of a frame-shifting deletion in a gene so named ALG5. And so we extended the familial study, and this variant was confirmed in 9 affected individuals, and it was absent in the unaffected individual. And so you can see that the patients do have non-enlarged polycystic kidneys. And to search for additional pedigrees, we screened a large cohort of ADPKD, ADPLD, and ADTKD individuals, and also through collaboration with John Sawyer and Eric Ollinger, we explored data of 100,000 genome project. In total, we identified 23 individuals from 5 pedigrees. And so a few patients did reach kidney failure between 62 and 80 years of age. You can see here 2 pedigrees, and they tend to have a quite consistent presentation with atrophic polycystic kidneys and EGFR loss usually occurring later in life after 60 or 70 years of age. So ALG5, unlike most of the other genes we spoke about, but ALG5 had not been reported in human disease, including in recessively inherited disorders. It encodes here the dolichol phosphate beta-glucosyl transferase, which catalyzes the synthesis of a glucose donor. And this is required for the addition of the 3 glucose residues on the top of the N-glycan chain. And so I told you that it was very functionally very important to have these 3 glucose. And so we hypothesized that the ADPKD-like phenotype observed in the 5 pedigree of individuals harboring ALG5 variants might result from a glycosylation defect leading to an intermaturation of proteins, including notably PC1. And in the interest of time, I will have to skip this part here, but thanks to a collaboration with François Fouquier, who is a glycobiologist in Lille, we were able to show that ALG5 deficient cell and ALG5 cells with only 1 copy of ALG5 were in fact had a defective glycosylation, and with an accumulation of immature form and no mature form of the N-glycan chain, either before or post-transfer. And then what Hugo Lemoine, PhD student in my lab in Louvain-Roux, PhD showed is that the loss of ALG5 specifically affects PC1 expression and maturation. We were able to show that there was a loss of the mature form of PC1, and then retransfected the wild type form, but not the variants the construct with the variants identified in the patients, was able to rescue the PC1 maturation profile. Loss of ALG5 also impairs holistin localization, and here we can see that in ALG5 deficient cell there is no membranous expression of PC1 and no detection of the PC1-PC2 complex on the cilium. So here is a kind of mugshot gallery of all the different forms of ADPKD. Recently, the KDIGO guidelines published just in 2025 emphasized the importance of a precision diagnosis in ADPKD and promote the idea of a gene-based nomenclature where you would add ADPKD plus the gene name whenever it is known. When genetic testing is not performed, is not accessible, is inconclusive, or is negative, you would still go with ADPKD, but each time you have the information, instead of saying just ADPKD, use ADPKD PKD1, ADPKD PKD2, or ADPKD IFT140, etc. And so this is a summary of the respective distribution of the different etiology, at least in clinical cohorts. So beyond these etiologies, there are several ADPKD phenocopy, and I will just show them and list them in the interest of time, because you can see that it's time now to move on to the Q&A part. But some patients might be considered initially as being affected by classical form of ADPKD and later recognized as having, for example, orofaciodigital type 1 disease, which would be, it's a rare differential diagnosis in notably in women, because it's in most cases embryonically lethal in male, but it can really be missed, although normally there would be some distinctive extrarenal features. Some patients are not correctly recognized, and this also emphasizes the importance of a precision diagnosis. And so I will go. I will skip this part just to have time for the questions. So in conclusion, I would like to say that genetic testing in ADPKD patients provide diagnostic and prognostic information and can help to reclassify the different forms of ADPKD. This can have therapeutic implications. We mentioned, for example, that there is no evidence for efficacy, tolvaptan efficacy in non-PKD1, non-PKD2 forms of ADPKD, and in fact there are more and more clinical trial ongoing in ADPKD, and this also emphasizes the importance of a precision diagnosis. 5% of individuals with a diagnosis of ADPKD have a causative variant in all the genes, PKD1 or PKD2. There are a total of 20 different genes in so we discussed the different genes associated with the ADPKD spectrum, and then you have several genes involved in differential diagnosis. And it's important to keep in mind that imaging-based diagnostic and prognostic tool cannot be employed in this patient. The gene-based nomenclature in ADPKD, promoted by the KDIGO guidelines, aim to provide a better appreciation of the expected phenotype and outcome for each specific etiology. And last, I think it's very important to emphasize the collaboration between nephrologists and genetic molecular geneticists and genetic counselor, clinical geneticists. And the way we do it is to work through a multidisciplinary board meeting, and this is a screenshot of our weekly board meeting, where we consider the genetic result together with the imaging result, the familial information, and it is, I think, of value because a lot of cases are quite complex. So I would like to thank all the colleagues and collaborators. Thank you for your attention, and I will be happy to address any question.
Thank you very much, Emilie, for this fantastic and really impressive presentation. Before we take the questions, I'd like to remind you to put your questions in the question box or in the chat box, so I can read them out. So far, I see only one question. Give you another minute while I'd like to announce the next webinars. The next regular webinar will be on the 27th of January with insights into pathophysiology of idiopathic nephrotic syndrome, given by Felicitas Hegel from Hamburg. But there will be 2 additional webinars. The first one is on the 21st of January, which is for the first time a joint ERN webinar from several different ERNs, and it will be on a very topic, the device shortage we're all experiencing, that will be discussed there. And one day later, on the 22nd, there's a new format which is called the ERKSperience, which is a case-based discussion forum where we will start with the topic of hyperoxaluria. So those are the next webinars, and that we have one question here, which is the kind of obvious question he put in from German Munoz, who's asking whether or not we should do genetics in any polycystic kidney disease patient right now.
I would say yes, at least in every family. And I'm not here. I recognize that in some countries access can be difficult. In other countries, access is possible. In some countries, there is also the effort of getting it covered by insurance that might be a barrier. But the clinical, my point is that the clinical utility is extremely important for several reasons, and I've listed them here. But today we have prognosis evaluation, we have understanding sometimes atypical or very early onset forms. We have the other indication of preparing possibility of reproductive counseling, because, so I'm not speaking about the minor gene, but in patients with severe ADPKD, PKD1, for example, discussing the possibility of implantation, genetic testing will necessitate to have identified the variant. Section of living kidney donor or differential diagnosis. So all these are today's indication. And then on tomorrow, well, there are some specific therapy being developed. So I've listed just here a small example of chaperone therapy, which is really variant specific, well, domain specific. So this is an ongoing trial evaluating a chaperone in a specific domain of PKD1, of polycystin-1, but there might be other therapies that will be gene dependent or domain dependent or allele specific. I think it's very important to have the cohorts of patients with genetic testing already performed for clinical trial readiness. So this is another consideration, but all the other indications are on the left side do apply to a lot of our patients. So I would say, except the situation of a patient who has already reached kidney failure, who do not have any relative who would have benefit of the result of the genetic testing, I think that having a genetic testing is important for all of the others.
All right, thank you very much for that. They're very important. If I may ask, we have seen a lot of patients and pictures from patients with a massive cyst load in their kidneys in these atypical ADPKD forms, and I was wondering, patients apart from kidney function decline experience other problems like bleeding, cyst bleedings, or cyst infections? Is that kind of the same as in ADPKD, or does that differ?
In IFT140 patients, some do have cyst-related complications, such as pain or bleeding. or infection, because the cystic burden can be quite important. But a majority of the patient would remain under the radar. But I would say mostly with IFT-140, because some do have really large cysts. They have a part of the cohort, I don't remember exactly the percentage in our clinical cohort again, but some did have previous either cyst fenestration or cyst aspiration for example, pain, because there was a 12-centimeter cyst having a compressive effect. But apart from that, there are less cyst-related events than in classical forms of ADPKD.
Okay. Then there's a question from Lucas Bergerhausen, who's actually sitting next door for me here, who thanks you as well for the fantastic presentation, and who's asking, as tolvaptan is not an opportunity in these patients, does the genetic results have an implementation or a consequence for any kind of treatment? Is there any compound?
No. We say that genetic testing, for example, if we take again the example of IFT-140, it's a situation in which genetic testing can bring good news. So that's a first good argument to perform it, because you can reassure the patient on the overall good prognosis. I've seen one patient in whom tolvaptan was discontinued because it had been, it was not a patient from our center, but tolvaptan had been introduced because the patient had a large TKV, but in fact, again, it was layer 2A, because it was a large TKV indeed, but a small number of cysts with, and so then we advised to discontinue, and not having to be under tolvaptan was also good news for the patient. I mean, it's extremely important, and I'm totally pro tolvaptan when there is an indication, but if there is no expected benefit, it's better to avoid it. But there is no specific treatment for any of the forms, so most of the other forms you saw have a good prognosis anyway. But for example, for NEC8, which has, as you saw, a bad prognosis, there is no specific treatment so far. But recognizing those forms is a first step toward a specific therapeutic approach later on.
Mm-hmm. And if I have seen it correctly, in the Jenkins cohort, there were about 260 patients genetically unsolved.
Yes. Yes, it's still a work. Yeah. Sorry.
Sorry, I was just wondering, what would you expect to find? Are there other genes not detected yet? Are there intronic variants in already known genes? Is it all epigenetics? What would you expect?
I think that so most of these cases, because I showed this was a split for individual patients, if I'm correct, and then in families it was a bit no, it was the split, sorry, for families, because most of these cases were sporadic cases, not a lot. We still do have a few unresolved families, and in that case I think we may be missing indeed some non-coding variants, and we are looking for those currently. There might still be additional genes to identify, but well, I'm not sure we will find. So we will not find a single genetic etiologist for. It will be the genes that are now identified. And we have another gene that I did not speak of because it's not published yet, but it's also idiopathy-associated gene. And what we see is that mostly low penetrance, mild presentation. So when it's really classical ADPKD, it's probably an intronic variant that is missed. And in the sporadic case, what we could be missing also are the mosaicism, because so they can be difficult to identify in blood DNA, even when we do specific analysis, they can be quite challenging to identify. So that would be my guess for some of those, at least.
Fantastic. As far as I don't see any further questions in the chat box, it only leaves me with thanking you again for that fantastic presentation. And yeah.
Well, thank you very much, and thanks again for the opportunity to present today. Thank you to all the attendees, do not hesitate to reach out if there are additional questions. Thanks again.
This is a great offer. Thanks a lot. It was great having you, and hope to see you all back on the 21st of January for our next webinar then. Thank you very much, and good afternoon.
Thank you.
All right.